Students, today we'll discuss about a very important topic, that is gene regulation. And in gene regulation, more specifically, that is your LacOperon, that is there in your syllabus. That is core 12, molecular biology, unit 5. So coming to gene regulation of gene, or you can say gene expression, it includes a wide range of mechanism adopted by a cell to regulate the production of a specific product by activating or deactivating specific genes. That means by switching on and switching off of a particular genes to get a particular product, right? Then coming to the LacOperon actually it is an inducer operon, and it is seen in prokaryotic cell, that is E. coli. That gets switch on and switch off according to the presence or the concentration of glucose, taking into consideration the concentration of glucose and lactose. So with response to this glucose and lactose concentration, this particular mechanism gets on and off. And this was, first of all, Jacob and Monod first of all originated the idea in 1961, and for this they shared the Nobel Prize in medicine in 1965. So, and what is an operon? Operon actually is a cluster of you can see operon is a cluster of genes, you can say. So these are the cluster of genes whose expression is controlled and coordinated by a single promoter. This is a promoter. This cluster of genes are controlled by a promoter, and which of course the promoter is, the first part is your operator, right? So this is actually, these are the structural genes, and these are the promoter and the operator. And the last portion of promoter, there is a cap site. This is your cap site, and just the upstream. So this is your upstream and this is your downstream. So the upstream to your cap site, there is a place which is known as your LAC inhibitor, and there is a promoter for LAC inhibitor, which in normal case, when there is no you can say lactose, this LAC inhibitor produces, or it translates, or it gives rise to a particular inhibitor protein, that is a repressor. So that repressor gets attached with the promoter, so that your RNA polymerase cannot move. So if it is being attached, then this cannot move, so it blocks. So that normally happens. So this is one way of controlling. So in the another way of controlling is your presence of by the attachment of cap in the cap site. So this cap is a protein. This is you can say this is a protein. This is a, you can say, catabolic activator protein. So this catabolic activator protein, this gets attached with your cap site, just which is upstream to your promoter, and when it gets attached, when there is no, or very, you can say, there is no glucose. When there is no glucose, so it gets attached with this cap site, and for that it needs another you can say molecular messenger, that is your cyclic AMP. So this cyclic AMP is being produced by the ATP, by the degradation, or by the catabolic, you can say by the enzymatic activities of the ATP by adenyl cyclase. So the ATP is converted, it is broken down to cyclic AMP, and this cyclic AMP, when it combines with your cap, it gets attached with the cap site. So what happens if it gets attached with the cap site? If it gets attached with the cap site, it helps the RNA polymerase to bind tightly with the promoter, so that they can have a high rate of transcription. So there are two methods of controlling this gene. One is by your LAC repressor. When there is no lactose, the LAC repressor will, and definitely it will combine with your promoter, so that it will inhibit the your RNA polymerase, so that there will be no transcription. And in the second, if the cap binds with the cap site along with the cyclic AMP, then it helps in the transcription in a high rate. So these are the two methods actually. So actually what happens, so if there is no sugar and there is lactose, this is the most ideal condition where it functions. So this LacZ, LacY and LacY, they will be transcribed to form an mRNA. It is a polycistronic mRNA, and this LacZ will produce beta galactosidase. This beta galactosidase, otherwise it converts the lactose to glucose and galactose, and to some extent it forms this allolactose. So allolactose is what? So if this, that means if there is lactose, means there are, you will find this allolactose. Allolactose is the isomers of lactose. So this isomers of lactose helps in getting attached with what is that, in your this repressor. So this repressor, it changes its structural it helps in changing the structure so that it can no more attach with the operator, so it comes down. So this, when the lactose is present, that means there will be allolactose, and allolactose are nothing but they are the inducible inducers. They are the inducers, and that is why it is otherwise known as inducible operon, right? So that is why it is otherwise known as inducible operon, and they are the inducers. So normally this is the condition where how this your LacOperon performs. Then coming to your next part is there are 4 conditions. Now we can come across with 4 conditions. That is when glucose is present and lactose is absent. So when the glucose will be present and lactose will be absent, so what will happen? Naturally there will be a repressor, so there will be no transcription. So when there is no lactose, like lactose is absent, naturally there will be a repressor, right? So that what will in the next, what is the glucose present and lactose present? So if the glucose will be present, naturally there will be no this caps and AMP formation, there will be no cap and AMP formation. But if there is lactose present, naturally this allolactose, which will be attached with this repressor, it will help to detach it from the operator. So what will happen? Now there will be transcription. So transcription can happen. But what is wrong is that, or what is the demerit here? It is that there will be a weak transmission, or it will be a slow transmission. So your synthesis of your different type of the substances from LacZ, LacY and LacA will be less amount. Why? Because the binding of this RNA polymerase with the promoter will be not that tight, so it will not help in a proper transmission, proper transcription, right? So this is the first condition, this is the second condition, and then coming to your third condition. So third condition is this is your third condition. So in the third condition, this is here what is there? The glucose is absent and lactose is absent. So when the lactose will be absent, then definitely there will be a repressor, there will be attached. And when the glucose is absent, so naturally this cap and AMP will be attached with the cap site, so for the helping of for the tight binding of your RNA polymerase with the promoter to help in the transcription, but there will be no transcription because here there is a blockage. So when glucose is absent and lactose is absent, the transcription cannot happen because there is a blockage here by the repressor, right? So in the fourth one, this is the fourth condition. So in the fourth condition, glucose is absent and lactose present. So this is the most ideal condition, right? So when the glucose is absent, and naturally the cap and cyclic AMP, they will be formed, they will bind with the cap site to have a stronger bondage with the promoter, so that there will be high level of, there is chances of high level of transcription, that is there, one is correct. So the second part is when there is sufficient amount of lactose are present, there will be allolactose, and the allolactose will bind with the repressor, and so that it will change its configuration, so it can no more attach with the operator. So naturally this RNA polymerase will have a transcription. This is the high level of transcription. This is the, you can see, the best way, or it is the ideal condition where the transcription takes place, or your lac operation happens. That means the glucose is absent. So when the glucose is absent, there will be a binding of the cap on the cap side or the catabolic activator proteins, and as because there is lactose is present, so naturally there will be it will act as an inducer, and it will combine with your repressor, and it will induce, or it will change its configuration so that it will be detached. So why it is called an inducer? It is because it induces the rate of you can say the transcription by the high concentration of the lactose. That means when the lactose is more, then it will induce to form the, you can say, the lactose. So we already know what are the different type of enzymes. So LacZ produces beta galactoside for the formation of your, you can say, this glucose and galactose, and the second is LacY. They form in the lactose permease, the formation of lactose permease. So lactose permease means it helps for the absorption of more amount of lactose into the cell, so that the cell can get, or the E. coli can get more amount of lactose. And the third one, the LacA, this enzyme is responsible, this gene is responsible for your thiagalactoside transacetylase. So actually it helps in the detoxification of your cell. So this is the normal process of, you can say, this LacOperon. Then coming to your the TRP operant. So TRP operant is just reverse. What is that? It is a repressive operant. It's a repressive operant, right? So the LacOperon is an inducer type, but it is a repressive type. So it is activated. So actually TRP operant means it helps in the formation of tryptophan. Tryptophan, actually this tryptophan helps in the formation of a particular, actually this mechanism actually helps in the formation of a tryptophan biosynthesis enzyme. So this your A, you can say, it is having a structural gene like A. It is structural gene like A, B, C, TRP D, E, and there is an operator and there is a promoter, and this is a RNA polymerase, we know. So actually tryptophan is it is an amino acid. The E. coli normally it gets this tryptophan from the environment, but when there is in the surrounding there is less amount of tryptophan or no tryptophan, so then naturally this the E. coli will require, or it will trigger the formation or the synthesis of tryptophan from its own cell, and so for that it needs to switch on these 5 different type of genes, and this is being controlled by a promoter and a operator. So normally what happens, it is a repressive type of operant, because normally when there is sufficient amount of tryptophan, so this tryptophan joins with the repressor molecule, to combine with the operator molecule, so that to inhibit the translation, inhibit the transcription. So this transcription is inhibited in normal condition when there is sufficient amount of tryptophan. Why? Because this tryptophan acts as a co-repressor. Actually 2 molecules of tryptophan, they gets attached with the repressor protein, and this now it changes its configuration and it can get attached with the operator, right? So now if it is getting attached with the operation, it forms a complex, and this will inhibit the transcription process. So this transcription process will be inhibited because there is a, you can say, attachment of your repressor, and they perform these tryptophan molecules, they act as a co-repressor. So this is negatively regulated, and that is why it is known as, these are known as negative regulators. So when there is sufficient amount of tryptophan, it will inhibit. So normally it inhibits, in normal condition it inhibits, that is why it is known as repressive operation, the repressive operon. So when the tryptophan will not be there, so when there is less amount of tryptophan or no tryptophan, you can say in this condition you see that there is no, you can say, this there is no tryptophan here. So that is why this area is free. So now when this co-repressor is not there, it can no more attached with this operator, so that it detaches, and now your transcription can happen freely, right, to form the formation of, you can say, your your formation of tryptophan synthesizing enzyme, and ultimately to produce tryptophan. So this is the basic difference. These are very simple, you can say. This LacOperon is a inducer type, that means when lactose is there it will induce, and this tryptophan is just reverse. When the tryptophan is there sufficiently, it will inhibit, but when the tryptophan is when there is no tryptophan, so it can induce, or it can switch on this transcription process, because it can, this repressor can no more be attached with the operator, and now this the required amount of, you can say, this amino acids will be formed, and ultimately this enzyme will be formed. So thank you, students. This is all about the LacOperon classes.